Welcome to ScienceStars. Topic: Biotechnology and its applications. We are going to start a super fast revision for the upcoming boards exams, so let's get started. Critical research areas of biotechnology are providing the best catalyst in the form of microbes or pure enzymes. Secondly, creating optimal conditions for catalysts. Thirdly, downstream processing. Agricultural applications. At first, it made crops tolerant to abiotic stresses such as cold, drought, salt, and heat. It reduced use of chemicals and hence made pest-resistant crops. Reduced post-harvest losses. Increased efficiency of mineral usage by plants, which helps in sustaining fertility of the soil for a long time. Enhanced nutritional value. For exam, golden rice is vitamin A enriched rice. Important point to be noted that GMO or genetically modified organism with modified and manipulated genes. Bt toxin. It is produced by Bacillus thuringiensis, which kills insects such as lepidopterans, dipterans and coleopterans. It is initially present as protoxin, that is the inactive form, but is converted into active toxin by the alkaline pH of the insect gut. These toxins bind to midgut epithelial cells and cause the lysis and death of the insect. Important examples are Bt cotton and Bt corn. Specific Bt cotton genes are incorporated into several crop plants and are coded with the gene Cry. Example: Cry1AC and Cry2AB control cotton bollworm, and Cry1AB control corn borer. Pest-resistant plants. Meloidegyne incognita is a nematode parasite of the roots of tobacco plant and decreases its yield. Rnai, that is RNA interference mechanism, helps the plant to prevent its desiccation. Rnai occurs in all eukaryotic cells as a method of cellular defense. It produces both sense and antisense mRNA, thus making a double-stranded RNA and thereby preventing its translation, that is called silencing. And with the help of this method, it have been achieved to eliminate the parasite from the roots of tobacco plant. Human insulin, also known as humulin. Earlier, insulin was extracted from slaughtered cattle and pigs, but that caused humans to develop allergy-like symptoms. Human insulin consists of two short polypeptides chains linked with disulfide bridges, chain A and chain B. It is synthesized as a prohormone, and proinsulin has an extra stretch of a polypeptide known as chain C, which needs to be removed before maturation. In 1983, an American company, Eli Lilly, prepared chain A and chain B of human insulin using recombinant DNA technology in Escherichia coli bacterium. Isolated the separate chains and joined them by creating disulfide bridges. Gene therapy. First gene therapy was given to a 4-year-old girl with adenosine deaminase deficiency ADA in 1990. This deficiency is caused by absence of ADA-cDNA or non-functional ADA-cDNA. This weakens the immune system. In gene therapy, lymphocytes are taken out of patient's body and cultured outside. Then a functional ADA-cDNA is added, and then the cells are returned to the patient. It helps in overcoming the deficiency. But as the lymphocytes are mortal, the patient requires periodic transfusion of cells. If bone marrow transplant is done at embryonic stage, it could be a permanent cure. Molecular diagnosis. Recombinant DNA technology, PCR and ELISA help in early diagnosis. PCR is routinely used to detect HIV in suspected AIDS patients. ELISA is based on antigen-antibody reaction. Transgenic animals. Animals that have a foreign gene expressed are called transgenic animals. Common reasons to produce transgenic animals include: to study how genes work, to study disease such as cancer, Alzheimer's, etc. For production of biological products, for example, APLA1 antitrypsin is used to treat emphysema. First, transgenic cow-produced human protein-enriched milk. To test vaccine safety. Chemical safety testing. It involves procedure same as quality testing or toxicity testing of drugs. Ethical issues. GEAC stands for Genetic Engineering Approval Committee. It makes decision regarding validity of GM research and safety of GM organisms for public services. Biopiracy is the use of bioresources by multinational companies or organization without authorization from countries or people concerned. For example, the case of basmati rice and turmeric neem. Comment your doubts. Don't forget to subscribe the channel and share it with your friends.